Placentation. In biology, placentation refers to the formation, type and structure, or arrangement of the placenta. The function of placentation is to transfer nutrients, respiratory gases, and water from maternal tissue to a growing embryo, and in some instances, to remove waste from the embryo. Placentation is best known in live-bearing mammals, theria, but also occurs in some fish, reptiles, amphibians, a diversity of invertebrates, and flowering plants. In vertebrates, placentas have evolved more than 100 times independently, with the majority of these instances occurring in squamate reptiles. The placenta can be defined as an organ formed by the sustained apposition or fusion of fetal membranes and parental tissue for physiological exchange. This definition is modified from the original Mossman 1937 definition, which constrained placentation in animals to only those instances where it occurred in the uterus. In mammals, in live-bearing mammals, the placenta forms after the embryo implants into the wall of the uterus. The developing fetus is connected to the placenta via an umbilical cord. Mammalian placentas can be classified based on the number of tissues separating the maternal from the fetal blood. These include Hemochorial placentation. In this type of placentation, the chorionic villi are in contact with the endothelium of maternal blood vessels, e.g. in most carnivores like cats and dogs. Hemochorial placentation. Chorionic villi, growing into the apertures of uterine glands epithelium, e.g. in ruminants, horses, whales, lower primates, dugongs. Hemochorial placentation. In hemochorial placentation, maternal blood comes in direct contact with the fetal chorion, which it does not in the other two types. It may avail for more efficient transfer of nutrients, etc., but is also more challenging for the systems of gestational immune tolerance to avoid rejection of the fetus, e.g. in higher-order primates, including humans, and also in rabbits, guinea pigs, mice, and rats during pregnancy. Placentation is the formation and growth of the placenta inside the uterus. It occurs after the implantation of the embryo into the uterine wall and involves the remodeling of blood vessels in order to supply the needed amount of blood. In humans, placentation takes place 7–8 days after fertilization. In humans, the placenta develops in the following manner. Chorionic villi from the embryo on the embryonic pole grow, forming chorion frondosum. Villi on the opposite side of embryonic pole degenerate and form the chorion leaf or chorionic levy, a smooth surface. The endometrium from the mother over the chorion frondosum. This part of the endometrium is called the decidua basilis, forms the decidual plate. The decidual plate is tightly attached to the chorion frondosum and goes on to form the actual placenta. Endometrium on the opposite side to the decidua basilis is the decidua parietalis. This fuses with the chorion levy, thus filling up the uterine cavity. In the case of twins, dichorionic placentation refers to the presence of two placentas in all dizygotic and some monozygotic twins. Monochorionic placentation occurs when monozygotic twins develop with only one placenta and bears a higher risk of complications during pregnancy. Abnormal placentation can lead to an early termination of pregnancy, for example, in preeclampsia. In lizards and snakes, as placentation often results during the evolution of live birth, this means that the occurrence of placentation in squamata is more frequent than in all other vertebrates combined, making them ideal for research on the evolution of placentation and viviparity itself. In most squamates, two separate placentae form, utilizing separate embryonic tissue, the choriolentoic and yolk sac placentae. In species with more complex placentation, we see regional specialization for gas, amino acid, and lipid transport. Placentae form following implantation into uterine tissue as seen in mammals, and formation is likely facilitated by a plasma membrane transformation. Most reptiles exhibit strict epithelial placentation, e.g. Pseudomoia. Antracastoia, however, at least two examples of endothelial placentation have been identified: Mabuia, sp. and Trachypes ibisi. Unlike eutherian mammals, epithelial placentation is not maintained by maternal tissue, as embryos do not readily invade tissues outside of the uterus. Research. The placenta is an organ that has evolved multiple times independently, evolved relatively recently in some lineages, and exists in intermediate forms in living species. For these reasons, it is an outstanding model to study the evolution of complex organs in animals. Research into the genetic mechanisms that underpin the evolution of the placenta have been conducted in a diversity of animals, including reptiles, seahorses, and mammals. The genetic processes that support the evolution of the placenta can be best understood by separating those that result in the evolution of new structures within the animal and those that result in the evolution of new functions. Evolution of placental structures. In all placental animals, placentas have evolved through the utilization of existing tissues. In viviparous mammals and reptiles, placentas form from the intimate interaction of the uterus and a series of embryonic membranes, including the choriolentoic and yolk sac membranes. In guppies, placental tissues form between the ovarian tissue and the egg membrane. In pipefish, placentas form following the interaction with the egg and the skin. Despite the placenta forming from pre-existing tissues, in many instances new structures can evolve within these pre-existing tissues. For example, in male seahorses, the underbelly skin has become highly modified to form a pouch in which embryos can develop. In mammals and some reptiles, including the viviparous southern grass skink, the uterus becomes regionally specialized to support placental functions, within each of these regions being a new specialized uterine structure. In the southern grass skink, three distinct regions of the placenta form which likely perform different functions. The placentome supports nutrient transfer via membrane-bound transport proteins, the paraplacentome supports the exchange of respiratory gases, and the yolk sac placenta supports lipid transport via apocrine secretion. Evolution of placental functions. Placental functions include nutrient transport, gas exchange, maternal-fetal communication, and waste removal from the embryo. These functions have evolved by a series of general processes, such as repurposing processes, found in the ancestral tissues from which a placenta is derived, recruiting the expression of genes expressed elsewhere in the organism to perform new functions in placental tissues, and the evolution of new molecular processes following the formation of new placenta-specific genes. In mammals, maternal-fetal communication occurs via the production of a range of signaling molecules and their receptors in the choriolentoic membrane of the embryo and the endometrium of the mother. Examination of these tissues in egg-laying and other independently evolved live-bearing vertebrates has shown us that many of these signaling molecules are expressed widely in vertebrate species and were probably expressed under G. This suggests that maternal-fetal communication has evolved by utilizing the existing signaling molecules and their receptors, from which placental tissues are derived. In plants, in flowering plants, placentation is the attachment of ovules inside the ovary. The ovules inside a flower's ovary, which later become the seeds inside a fruit, are attached via funiculi, the plant part equivalent to an umbilical cord. The part of the ovary where the funiculus attaches is referred to as the placenta. In botany, the term placentation most commonly refers to the arrangement of ovules inside an ovary. Placentation types include basal. The placenta is found in mono to multicarpellary, syncarpous ovary. Usually a single ovule is attached at the base bottom, e.g. helianthus, tridex, tagetes. Parietal. It is found in bicarpellary to multicarpellary syncarpous ovary. Unilocular ovary becomes bilocular due to formation of false septum, e.g. cucumber. Axile. It is found in bicarpellary to multicarpellary syncarpous ovary. The carpels fuse to form septa, forming a central axis, and ovules are arranged on the axis, e.g. hibiscus, lemon, tomato, lily. Free central. It is found in bicarpellary to multicarpellary syncarpous ovary. Due to degradation of false septum, unilocular condition is formed and ovules are arranged on the central axis. E.g., dianthus, primula primroses, marginal. It is found in monocarpellary unilocular ovary. Placenta forms a rigid along ventral side and ovules are arranged in two vertical rows. E.g., Casum sativum p.